Alright, so in this video I'm going to talk about the functions of an antibody. So let's just go ahead and review what an antibody is. An antibody is a B cell receptor that has been excreted. Right? So essentially, you have a B cell, and on the surface of the B cell is this little receptor, and it's called, when it's in the B cell, it's called immunoglobulin, and it's frequently abbreviated like this, right? And remember that they have this end that is called FC, or the constant fragment. So for every single B cell receptor for that specific person, this end is going to be exactly the same. And then on this end of the immunoglobulin, or the B cell receptor, there you have the variable region, and this is where we get that sort of random genetics that allows for a high degree of specificity in the hands here. I like to call them the little hands, right? That will grab onto a specific antigen. And every single one is a little bit different and will respond to something that comes into the and some antigen that comes in from some foreign source, right? So what the B cell receptor, or what the B cells do, is after the receptor has, say, responded to some sort of antigen, it triggers that B cell to start making antibody, start making copies of itself, which are called plasma cells. So plasma cells. Right. And it starts to pump that antibody out, and the antibody then drains from the lymph nodes. Remember that the B cells stay in the lymph nodes, but the antibody will drain from the lymph nodes and go to the site of infection and will perform several different functions. So remember that the antibody is then going to have these very specific hands and this constant fragment on the end. So there are 4 different functions of what the antibody does when it drains to the site of infection. The first one is neutralization. All right, so let's say we have some big compound, I don't even know, it could be like a toxin. So let's say this is a bacterial toxin or a viral toxin or something, and it binds to neurological receptors in people, and does some sort of neurological damage or causes some sort of prevention of nerve function, right? Well, if an antibody recognized a part of this compound and came in and was able to bind to it, right, by grabbing on with its little hands, that would prevent the function of that toxin. So it essentially neutralizes the toxin and prevents it from being able to cause that neurological disruption. We could also neutralize a piece of DNA, or you could bind to a whole cell that prevents maybe some toxin from being pumped into or out of the bacterial cells. There's a lot of different things. So it's really just like it says, it neutralizes some part or prevents the function of some other compound or mechanism. So that's the first function of an antibody. The second is opsonization. So let's say that we have an E. coli here, or bacterial cell, and we have an antibody that is attached to the outside of it. Sorry, I keep drawing them different. So when you have the antibody attached to it, that's horrible, let's draw one again. Okay, this end, remember, is the constant fragment. So if we were to then have a much larger cell, a macrophage, come along, the macrophage will more readily engulf this bacteria because it recognizes that there is antibody attached to it, and it does it by recognizing the constant fragment. Because remember that macrophage and phagocytosis is a non-specific response, which means it's not responding to something specific about the foreign agent. It's just responding to the fact that there is something foreign. And so it's responding to this constant fragment being present. So opsonization is just when antibodies bind to microbes and trigger them to be engulfed or phagocytized by macrophages or neutrophils or other phagocytic cells. All right, so the third one is then triggering complement fixation. Right? So remember that the complement system is a process of building the MAC, which is the membrane attack complex, which is basically this cascade of protein manipulation in order to make, I like to call it the little protein donut, but it's a little pore channel or an opening that is drilled into the surface of a cell membrane. And if you drill enough holes or create enough protein donuts in the surface of a bacterial membrane, the bacteria will lyse. So if we have a bacteria here that has an antibody attached to it, that triggers complement fixation and the complement pathway. It's called the classical complement pathway. And so along we'll start to have, again, it recognizes this constant fragment. And so it starts to drill these little membrane attack complexes into the bacteria until it, allows stuff to travel in and out and it blows up and it lyses. All right. Now, the last function of an antibody at the site of infection is agglutination. And basically that's just saying that antibody acts like a glue. So where we have a site of infection, we don't really want the bacteria to be able to move freely through the body, right? We don't want them moved to another location. We don't want them getting into the bloodstream. So we want to stick them all together in one place so that we can come and we can, fight the battle, kill the bacteria, clean up the site of infection, and begin to get healthy again, right? So if I have a whole bunch of bacteria sort of at one place, how do I prevent them from moving from one place to another? Well, remember that the antibody has two different hands. So if you have one antibody here and it's holding on to antigen on this cell surface, and on this cell surface, it kind of locks them together, right? Then let's say I have another antibody here and it's locking this one, and then I have another one here, and this here, and then we've got, and it's locking this one together, and here, and here. All right, I'm epically failing at doing these drawings, but essentially what it does is it creates all these cross links sticking all those bacteria in together, preventing them from moving from one location to another. So then in can come macrophages and dendritic cells and neutrophils in order to fight even cytotoxic T cells to come in and attack and sort of eliminate this bacterial invader. All right, so those are the 4 functions of an antibody at the site of an infection.